have several things that we want to do before we introduce our keynote speaker, Dr. Mark Merchant, later in the program. But I do want to introduce a couple of special people who have a few words to say to the group. One individual is our board chair, Mr. Wayne Parker. One of the hallmarks of our board is strong leadership, and for many, many years we've had outstanding board leadership. And Mr. Parker is in his first year as board leader, and he is doing an outstanding job and just a real gentleman to work with. And so I'm very, very happy that Mr. Parker has ventured all the way from Northeast Louisiana to be with us today. Mr. Parker has actually served on the board since 2003. He's a graduate of Louisiana Tech University, where he majored in business administration and played football. Mr. Parker, please come forward. Thanks for being here.
Thank you, Brad. I'd like to have everybody's attention right now. Dr. Moffitt left me in charge, so if there's anything that anybody needs, welcome on behalf of the Board of Supervisors for the University of Louisiana system. It's a pleasure to be here with you all. I am so impressed with the work taking place at our universities, exemplified by the presentations that you're giving. What is exceptional about today is that it's not only an opportunity for students and faculty to showcase the high level of work you're doing, but to also exchange ideas with your peers at other UL system schools. Please know that your work is valued by our board, and we support the exceptional learning opportunities championed by our faculty and staff so that you, our students, can have meaningful experiences as you earn your degrees. Enjoy the rest of the academic summer.
Thank you, Mr. Parker, and we certainly appreciate your support and the support of our fine board. We're also fortunate to have with us today Dr. Catherine Watley, who's the Provost and Vice President for Academic Affairs at Berry College in Rome, Georgia. Dr. Watley is a nuclear physicist and has been an active participant with the Council of Undergraduate Research since 1987, including organizing 3 national conferences. The Council of Undergraduate Research is a national organization that was founded in 1978 and now represents over 900 colleges and universities around the nation. Its mission is to support and promote high-quality undergraduate student-faculty collaborative research and scholarship. And it believes that faculty members enhance teaching and contribution to society by remaining active in research and by involving undergraduates in that research. I'd like to thank Dr. Watley for being here and ask her to come forward to say a few words. Dr. Watley.
Thank you for inviting me. Thank you for inviting me to be here today. I have been going to talks this morning and have heard some really marvelous things. I'm very impressed with the performance of the students and the support of the faculty members and the administration of the system and of the schools to be here today and to help encourage students in these endeavors. I want to just bring greetings from the Council on Undergraduate Research and talk just for a minute about the differences and the similarities between 2 organizations that have now merged. The Council on Undergraduate Research and the National Conferences on Undergraduate Research. The Council on Undergraduate Research is a national organization, as Brad just said, that is mainly oriented toward faculty. So it provides resources for finding out about where the grant opportunities are, for building alliances between institutions, for networking, and just general support of faculty who are involved in this endeavor of undergraduate research. And I can tell you from many, many years of experience, it's one of the greatest group of peers that you could possibly have. People share things, they are supportive of each other, they give you ideas, you can call them on the phone and say, I'm hitting this roadblock, what do I do now? Whether it's about your research or about just dealing with students or the best practices, all of that. The Council on Undergraduate Research is really wonderful at helping support faculty and provide resources for them for the undergraduate research endeavor. There's a website, cur.org, very simple, where you can go and look. Membership is not expensive. There are institutional members, or there are individual memberships, but there are also institutional memberships and enhanced institutional memberships, which are very affordable for institutions. And if you do the enhanced one, any member of your faculty can join for free. So that's a really wonderful system or institution-wide thing to look into. So talk to your, I know I'm talking to the provost here, so we'll talk about that some more. But anyway, it's a great organization. CUR holds a conference every other summer. There is one this summer. It's at the College of New Jersey in Ewing, New Jersey, and there's information available on the CUR website about that. The other organization, which has just joined CUR, is called NCUR, the National Conferences on Undergraduate Research. That started in 1987. I'm proud to be one of the founding members of that conference, and was on the original steering committee for that. It's an organization and a conference that focuses on students. So it's a place for students to come and present their work. It happens every spring. It just happened about 2 weeks ago, this past spring, and it is going to be next year at the University of Wisconsin-La Crosse. Students from all disciplines are welcome. They have poster and oral presentations. They also have gallery talks and other kinds of performances. It is for all disciplines: science, humanities, social science, fine and performing arts, business, anything. Anything your students are doing is welcome at NCUR. There is a fairly rigorous selection process. So you do actually get peer reviewed, but it's designed to be a conference where undergraduates are encouraged and supported, and again have a chance to present their work. So all of that now is covered in the envelope of CUR. You can find lots and lots of information on the website, but I can tell you right now, if the executive committee folks from CUR were here today, they'd be extremely pleased with what you all are doing today. Very, very impressive. So thank you very much for inviting me. I look forward to talking with you later.
Thank you, Dr. Whatley. We're fortunate certainly to have members of our council and individuals such as you from CUR to help guide us as we venture into these new waters called undergraduate research and scholarship. Faculty members in the audience will certainly want to make special note of an opportunity to hear more from Dr. Whatley about trying to grow and sustain undergraduate research and scholarship on our campuses at 1:15 p.m. at a faculty development session. That'll be held in the La Jeunesse room, which is situated in this complex. We'll help direct you to the La Jeunesse room at the conclusion of today's program. Now, at this time, in keeping with the focus of exceptional student work, it's only fitting that we recognize each of our student presenters with a new tradition that we've established with this new UL System Academic Summit. We have ordered and received some actually very, very nice UL System Academic Summit medals that we would like to award to each of our students. I'd like Mr. Parker and Dr. Phil Williams, President of McNeese, to join me on stage here. When your university's name is called, we would like the faculty mentors, president, provost, to head over to the area where the risers are. And when that university's name is called, those students that are either presenting this afternoon, whether it be through an oral presentation or a poster, or have presented this morning, please join me over here by the stairs. You'll walk across the stage and receive your medal. First of all, let's start with Grambling State University. Will the Grambling students please come up to my side here, and Grambling officials over to the risers. And as we're snapping the picture, would the folks from Louisiana Tech come to the front, please? State University. Northwestern State University. Southeastern Louisiana University. Lafayette. University of Louisiana at Monroe. University of New Orleans. Finally, our host from McNeese State University.
recognized internationally for his work in the field of alligator immunology. His research in alligator serum is integral in international research efforts that call for using crocodilian proteins for disease control. His work has encompassed isolating and determining the primary structure of peptides, characterizing the range of antimicrobial activities of peptides for a broad spectrum of pathogenic bacteria and fungi. Dr. Mergent has made several television appearances. He has appeared 4 times on National Geographic, once on the Discovery Channel. He's been on Canadian television networks, Good Morning America, Fox News in the morning, and his work has been highlighted in several documentaries. He has had over 65 publications. He has 6 more in the press and 8 that have been submitted over the past few years. These journals include Comparative Biochemistry and Physiology, the Journal of Experimental Zoology, Veterinary Immunology, Zoological Studies, and the Journal of Parasitology. Parasitology. How do you say that?
Administrator.
Thank you very much, Mark. The non-chemist. Dr. Merchant is frequently invited to make presentations on his research outside the states as well, in the other states as well as outside of the United States. He has presented his work in countries such as Australia, France, Brazil, Argentina, Bolivia, and Panama. When he comes to ask for travel, I never know where he's going. Not only is Dr. Mark Merchant an accomplished scholar and researcher, but he is also an excellent teacher. He continues to maintain the belief and practice that mentoring students is the primary goal of his work. Dr. Merchant is also the recipient of the 2007 McNeese State University Distinguished Professor Award. We are fortunate to have him on our faculty, and I'm pleased to introduce him today.
Good afternoon, everyone. It was almost 25 years ago. It was a mid-October day, 1987, and I was an undergraduate at Lamar University in Beaumont, Texas, just across the river here. I remember it was an afternoon, and I had completed one of my afternoon chemistry labs, and I was going down to my research lab. I packed everything up. I was heading out to the truck to go home. And I walked by the bank of faculty offices there in the Department of Chemistry. And there was a guy standing there that I was very familiar with, named Dr. Hugh Akers. I knew him very well because Dr. Akers at the time was he was a instructor for the biochemistry course in which I was enrolled. I knew him well because he had been my academic advisor for the last 3 years. And with all that, I had also been working in his undergraduate research lab for maybe 8 or 9 months. So I knew Dr. Akers very well. As I walked by, he asked me how I was doing. He was exchanging some pleasantries. He said, "You're supposed to graduate next May, aren't you?" I said, "I am." He said, "You got a second?" I said, "Sure." So he invited me to sit down. He said, "What are you going to do when you graduate?" Trick question. What's going on? So because I grew up in an area in southeast Texas, just like southwest Louisiana here, it's full of petrochemical industry. And when you're a chemistry major, you go get a job as a chemist. You're going to go that's just what you do. And so I told him, I'm going to be a chemist. He said, "Have you ever thought about doing anything else?" Not that there's anything wrong with that. I mean, have you ever thought about doing anything else? Like what? What else is there? He said, "Well, have you ever thought about going to medical school?" I think you can do medical school. Have you ever thought about a career in research? Because I used to be, I loved undergraduate research. It was nothing for me to be the only person in the building at 1 o'clock and 2 o'clock in the morning, once or twice a week sometimes, when I had time. And he said, "You seem to really like research," and he was right, I did. And So this conversation went on. He said, well, let me tell you. He said, no, don't shut any doors right now. It's still early. He said, why don't you take the NCAT, see what happens. Why don't you take the GRE, see what happens. See, maybe you might want to consider this stuff. Think about it. Don't close any doors. That's some good advice. Not just for academics, but I think leaving all your options open is good advice in lots of aspects of our lives. So I remember going home and thinking about it for a while. Okay, well, this guy, he knows a lot, he knows more than I do, so I scheduled myself to take the GRE, I scheduled myself to take the MCAT. And I remember I studied for these things, I took the GRE, and I think by the time I took the MCAT, I think I knew that I wanted to look for research, or at least pursue a PhD. And as I look back on my life, at the time when we were speaking, just that one afternoon, I had no idea the impact that 5-minute conversation was going to make. I mean, it really set my entire career. Without that conversation, I never would have considered going to grad school. I never would have considered a life in research. And I certainly, there's no chance I would be standing here in front of you today as an academic scientist. No way. So if you're an advisor, keep this in mind. You never can tell if you're a research advisor, if you're an academic advisor. You don't know what kind of small words are going to impact somebody's life. You just have no idea. I could probably point to several hundred students and wish I had never made that choice. Some of the ones I talked this morning probably rue the day that I talked to Dr. Akers 25 years ago. So with that being said, I want to talk to you about some of the research in which we are engaged here at Indiana State University. I've been here just over 10 years, and I've been really blessed with a good research program and great students in my lab, almost all undergraduate. You'll meet some of them toward the end. And I'm just going to explain one of our projects, the one that gets us most of the press, the one that people are most interested in talking about. And it's one that I'm interested in too. But we have many, many other projects. So I will start with why alligators. Most biochemists work with rabbits, mice, mammals. Alligators are very difficult systems to work with. There's no animal room in the country that can house alligators. Alligators fight really hard. And so I'm interested in alligators for a variety of reasons. I grew up in this area, a lot of hunting and fishing in the marsh. I've been around alligators my entire life, literally. But what interests me today as a biochemist is not the same thing that interests me when I was a kid. Whoops. Yeah. This is a photo I took in Bolivia a couple years ago. These are two caiman fighting. Crocodilians is a term I'm going to use several times. Crocodilian is a word that encompasses all 23 species of crocodile, alligator-like animals in the world today. They live in really great places. And all crocodilians, how come it's not going backward, Bill? Help me out. Backward button's not working. There, okay. All crocodilians are very territorial for members of their own species, and they fight. Obviously, like right now, alligator mating season is in full swing. It's just starting right now, and the big males are going off, and the females, 2 males cross, something like that's going to happen. And these guys have the equipment to do great damage to one another, obviously. And so what happens is, there's another picture I took in Belize about a year and a half ago. This the male on the left is throwing his, I got a pointer, is throwing his head back and he's pushing himself up. He's trying to look as big as he can. This guy's not pressed. And these two guys actually end up fighting. I've got a bunch of pictures of them blurry out of focus, just rolling around in the mud. I think that's a cool picture right there because that really shows some territorial posturing there that led up to a pretty good fight. Unless you were the one around, it wasn't that good. So these territorial disputes often end with this sort of result. They end in some serious injury, sometimes loss of entire limbs, sometimes one will kill the other. It's not uncommon to find these types of injuries. You can see this one's lost his left hand, this one's lost his entire right arm. Okay, he's pretty healthy. He's still got his tail to swim and still got his jaws to eat. Here's another alligator who lost about 3.5 feet of his tail. Interesting to note that these are not like lizards. They won't grow a tail back. They will not regenerate a tail. But it turns out that despite the fact that these animals live in swamps and muddy marshes and places where there's lots of opportunistic infections waiting to happen, that these enormous injuries basically heal most of the time without any sign of major infection. Now, if you cut my arm just a little bit, scratch it, put it in that water, it's going to get red and puffy and infected. These guys are moving entire animals out in the marsh and with limbs and no infection. Some people say, hey, that's pretty neat. I'm a biochemist. I say, hey, that's pretty neat. How's it work? And so we began to study the immune system of crocodilians about 10 years ago. And I'm going to take you through some of the things that we do. This project developed. It took about 2 years to get to this point. We started looking at alligator leukocytes, which are white blood cells. We know that at least for humans and mammals and birds and other animals, a good portion of the immune system is contained within the white blood cell. We have a lot of different immunological defenses that are contained within the white blood cells. So we started, I'm wondering, hey, is there anything in the alligator white blood cell that would be effective at killing bacteria? It's a reasonable question. Seemed like to me anyway. So we go out in the marsh, we go out in airboats, we go out in aluminum. It depends on the environment we're in. I catch alligators in a lot of different places, shallow water, deep water. Caught some last night. When we go out and we shine a spotlight, their eyes are very highly reflective, and you can see a big gator from 6 or 7 foot north away somehow, a long way, if you have a powerful spotlight. But you can see there's a noose around this alligator's neck. We put a noose around their neck, calm them down, bring them in a boat, tape their mouth up, and I can stick a needle in their neck. They have a large vein that runs out of the back of the skull here, and it runs the entire length inside the vertebral column, just over the dorsal to the spine, all the way down to the tail, inside the vertebrae. So I have to stick the needle in between the vertebrae. I can draw blood samples. So we take that blood sample back to the lab, and we take a look at some of the immunological things. Now, one of the things I do with this blood is I will isolate the white blood cells. So if you put an anticoagulant in the blood so that it does not clot, and you put the blood in these large tubes, this 50 milliliter tube, you can see the red blood cells are more dense. Same thing with humans, by the way, and they will settle by gravitation more rapidly than will the white blood cells. You can see right up here at the top, just at the top edge, there's no cells. That's the plasma. And then down here is most of your white blood cells, and of course all the red blood cells. So what I'll do is I'll stick a pipette in the top of this tube and kind of slurp up the white cells and let it settle some more during the course of the day between classes. Just keep on me and my students will slurp these white cells up, centrifuge them down. Now I have a big pellet of alligator white blood cells, which to me is gold. So we're going to take that alligator white blood cells and we're going to bust them open and get a very crude lysate of all the soluble stuff that's in a white blood cell. And we're going to do something like this. This is called a classical antimicrobial sort of inhibition. If you've ever taken any kind of microbiology class, you're familiar with this kind of thing. What I've done is I've taken a petri dish, taken some liquid medium bacteria food, basically a ground up cow in a bottle, and we take it, we sterilize it. Take that and we put just a little bit of bacteria. We see a little bit, inoculate a little bacteria in there. We pour it into a petri dish under a sterile hood. Let that solidify. And then we cut little plugs out, tiny, tiny little plugs. And then what I did was I took a little bit of that crude alligator leukocyte extract. I mean, just by a tenth of a drop, and put it in here. You put it in the incubator, you wait overnight, Shazam, you come back the next day, here's what you see. All this yellow material here is bacterial protein. If that wasn't there, it would be dark. Where I put these 4 and these 3 wells, where I put just a tiny little tenth of a drop of the alligator's leukocyte extract, white blood cell, busted up white blood cells, that stuff diffuses into this medium, and we have a place where it killed the bacteria, and thus the name of the zone of inhibition. It inhibited their growth in that zone. So we know that there's something in these white blood cells that are killing bacteria. Well, that's neat, but hey, that's not enough. We need to know what it is. So we want to figure it out. So what we did, the first thing I wanted to do is I did this sort of assay again. And I did it with a lot of different types of bacteria. That's just with one type of bacteria. So one of the things we did, we used a lot of different types of bacteria, and we measured the zone of inhibition. And it turns out in this original experiment, I think we tried, maybe 11, and 9 of them. We had activity against 9 of them. So what you're seeing here, the big bars, that alligator leukocyte extract had a big zone of inhibition. The small bars had a smaller one, but still somewhat effective. And of course, a couple of them had any effect. Tried the same thing on some yeast. These are all known to be pathogenic yeast strains. Candida is some yeast that are known pathogens in humans. But it turns out that when we tried on Candida yeast, I think 6 of the 8 species of Candida yeast were susceptible to whatever was in that leukocyte extract. And it turns out that this one right here, this Candida albicans, is by far the worst human health problem in the world, probably in terms of yeast. It is what MRSA is in the United States now, worldwide. Millions of people die from Candida infections, usually in third world countries, Africa and South America. So we got something in those extracts is really doing a number on these microbes. And we'd like to know a little bit more. We'd like to know what and how and why. So this is just a little mechanistic study we did, and I won't talk too much about it, but so when I take this leukocyte extract, basically we get 100% activity. If I treat it with something called a protease, it goes down to 0. That tells me as a biochemist, whatever it is in that extract that's killing these bacteria and fungi, it is protein. It's a protein. I can heat it to 70 degrees centigrade for 1 hour, which is quite hot. Most proteins will unfold and just ruin them. But when I cool it back down, I still maintain 100% of the activity. That tells me it's a little tiny little protein we call a peptide. And there's some other stuff. So we suspect at this point, through some other mechanistic studies, that we have what we call a polycationic antimicrobial peptide. It's a small protein that has multiple positive charges, and it has antimicrobial activity. So what are these antimicrobial peptides? Well, they're certainly, I'm not the first one to describe these. These things have been described in a broad spectrum of they've been isolated from a lot of different types of animals. They are surface active. If you look at the outside of a bacterium, it's full of phosphates, phospholipid membranes, it's a lot of negative charge. Well, my protein has positive charge. So it will bind the opposite charges. When it binds, it changes shape and it tears a hole in the membrane. So that it is active on the surface. A lot of antibiotics have to go inside the bacterium to be effective. This one's surface active. It's polycationic. I described that for you. So how are we going to figure out how to? This is what I suspect is in there. So this is the biochemical scheme that we have used to isolate these. And I'm not going to talk. It's pretty. Some of this is a little technical. We're going to bust open these alligator white blood cells in 10% acetic acid, which is a really strong vinegar. Vinegar is like 2.5–3% acetic acid. My lab always smells like acetic acid. The people around the lab office, they hate my lab because it always smells like dead bacteria or acetic acid. Then we're going to select. We're going to do something called ultrafugation. We're not going to talk too much about. We're selecting molecules between 1 and 10, like 1,000 molecular weight. We're going to do something called reverse phase high performance liquid chromatography, HPLC, and that's what we get. We've basically got 2 peaks here when I run this, and we separate all these proteins. And I don't want to talk. I know everybody's not a chemist, and good for you if you're not. So what we did was we isolated these 2 peaks. And I did an antimicrobial zone of inhibition assay with this one and with this one. The one on the left had no activity at all. The one on the right had really good activity. So we thought, we got it. We've isolated this protein. It's been 8 years. It took us. And then I did something which I like to call matrix-assisted laser desorption ionization time-of-flight mass spectrometry. Everybody. And so what this does, this tells you how big the protein is, how big that molecule is. So we got this protein. I want to find out how big it is. Is it in the range for most antimicrobial? And so I do this. I got 2 proteins in there. I thought I had 1 protein in it. There's 2 of them. One of them is that big, and one of them is that big. 40. They're all about 5 kilodaltons, a little bit less than 5 kilodaltons. now I got these two proteins and they're about the same hydrophobicity, they're the same size, they're both acid soluble. How am I going to separate these? Well, we did something called ion mobility chromatography, and we were able to, I'm not going to talk much about that, we were able to isolate these two proteins, and it turned out that was a good thing because we were able to then go on and determine the sequence. If you've ever taken biochemistry, you'll recognize that these letters each stand for an amino acid. The ones I've highlighted in red have positive charges, histidine, lysine, and arginine. You can see there are a lot of positive charges, a net charge of +8. This one has a net charge of +13, which is hugely positive. So we're on the right track, we think. In addition, they have these cysteines, these blue ones, I believe, the C. And so when I look at this structure of these peptides, they look an awful lot like a group of proteins that humans make called defensins. Defensins are small antimicrobial peptides. Everybody here has them. If you didn't, you probably would not be alive. They're that important. And if you see, there's a lot of similarities. Look at all the red and blue. There's a lot of similarities. We've done some alignment, and that's where we think we're heading. We think that we have isolated a new class of compounds from alligators. I think it's much more potent than many of the other peptides, antimicrobial peptides that have been isolated from various species. I think that alligators and crocodiles in general, crocodilians, have put a tremendous selective pressure on themselves to evolve something that's really good. Because if you're going to be a really aggressive animal, and you're going to have these territorial disputes, and you're going to fight to tear limbs off each other, you're either going to have to move to a cleaner environment. Maybe you can fight the clean room or something. Or you're going to have to stop that behavior. Or you're going to have to develop something like a really strong immune system that will allow you to tolerate that behavior. And I think that's what we're seeing here. It's a tremendous selective repression. So what are we doing now? Well, we want to synthesize these peptides, and we've done that. That wasn't easy. We want to do a computer modeling to get a three-dimensional structure. We want to look at antimicrobial peptides in other tissues. I have reason to believe that alligators' mouths and their esophagus are producing these as well. And I want to look at other crocodilian species, because you know what? Crocodiles are the coolest places in the world, let me tell you. We work with Cayman laterostris in Brazil. I've worked with Cayman jacare in Argentina. Costa Rica has the big saltwater, the American saltwater crocodile. Africa, that's a full-grown crocodile right there. That is the African dwarf crocodile. Beautiful animal with this really dark skin, really crazy. They're stupid. I can't believe they're stupid. That's a really neat animal. That's in Africa as well. This is the African slender-snouted croc. It has evolved. You can see how long and slender the snout is. It eats only fish. It's evolved to live in a river system. And it's difficult to tell because I've covered up most of the body because I'm so bulky. This entire animal is very slender. It has evolved to live in a riverine system, basically, and it's very slender, so it can be hydrodynamic. So that's an extremely fast animal, really, really hard to catch. Yeah, that's in Florida. That's more American crocodiles. Crocodilus morleti in Belize. That's in southern Mexico. That is Cayman crocodilus, and that's the big giant saltwater crocodile in Australia. So as long as I can keep those grant dollars flying, I can keep traveling. I've got the best job on the face of the earth. I'm trying to insult our provost, and I'm not sure. We'll have to negotiate something. But I think some of the best things about my research isn't necessarily the traveling I get to do. It's the people that I get to meet and the students with whom I have the opportunity to work. Farida, come back, Farida. Farida is now a pharmacist. Rodolfo is about to apply to medical school. Ross is in his third year of medical school. Clint is working on his PhD up at Georgetown. Paige she's writing her dissertation right now. Her PhD is in biochemistry. She's at University of Texas Medical Branch in Galveston. Jessica hides in the back. There's a medical school in Denver. Charles, he's about to apply to med school. Then Kata is working on his PhD at Mississippi State. Sarah was a smart one. Sarah married a guy who's in med school. And then these two ladies, Kylie Hurd and Stephanie Mead, are both in their third year of med school up in Shreveport. I could go on forever and ever. Brandon is finishing up his PhD at LSU in chemistry, and he's up at the University of Nebraska. I think he did third year of his PhD. And I can't tell you how satisfying it is to see these guys go on and succeed at the next level. That's one of the great pleasures I get. So I would like to I would be remiss if I didn't thank some of my funding agencies, and of course thank some of the people down at Rockefeller Refuge who have helped me collect alligators over the years, and also a couple of my colleagues over at LSU in Baton Rouge. And with that, I thank you for your time, and I would like to answer some questions if you have any questions about alligators or crocodiles or research. Or
We may have time for 2 questions.
okay, that's fine.
Anybody have a question?
If it's real hard, I don't know.
Get my exercise here.
Hi, thanks for an excellent talk, first of all. And then second of all, I noticed that there's, for the high number of cysteine residues, I know cysteine is a very rare amino acid, so from a biochemistry standpoint, what does that do for the mechanism of the protein?
It allows for disulfide bonds.
Disulfide bonds, but does that regulate the peptide unfolding, or is it?
Now, disulfide bonds add structural integrity, obviously. Oh boy, here I go with the biochemistry. There's 4 weak forces that hold proteins together. There's van der Waals forces, there's hydrogen bonding, there's dipole, all these. But the cysteine, the disulfide bond, is a covalent bond, and it adds a lot of structural integrity to the peptide.
One more question.
That was a little tough, man.
That was tough. That was above my pay grade.
I'm not checking my vitamins. I'm just making stuff up. Any more questions?
One question, yes.
Okay.
So you said the mechanism, the antimicrobial mechanism is ripping of the cell wall?
Yeah. These peptides tend to be very amphipathic. And so I didn't get into that, but what happens is you'll have one surface that is very positively charged, and another surface is very hydrophobic. So when it changes conformation, it dives down into the membrane. And so that's where the amphipathicity comes in, if I may be so bold.
So the bacteria that are resistant to it and not susceptible, what is their mechanism?
Yeah, I'm assuming that maybe they have different charge densities. Maybe they don't have the right charge. I'm not sure. That's a good question. And that is the assumption. And that's also the assumption why maybe alligators can make this and it doesn't affect their cells. Because eukaryotic and prokaryotic membranes look very different. The charge densities are good. We have a lot more positive charge in our membrane. Does that make sense? It's all about charge density. Remember that. That's another great piece. Now, before I stop, I have a friend. I told him. I just unplugged the mic or the cord. I'm not sure. But I had a friend that I told you I would introduce to you. Is it working? I'm not sure what I'm going to do with this now. What am I going to do? Okay. So I caught this alligator last night, and he heard so much about the academic summit he wanted to make a personal appearance. And there's something I would like to share with you about alligators, and some things that even people that are versed, and whenever you take an alligator in the back, you got to make sure the mouth is still taped shut. So I've got this alligator. I caught him near my home in Texas yesterday, and let's see here. He's not happy. He's not happy. They hiss as a threatening gesture, like a lot of women I know. See if I can. These are very strong animals. They're very aggressive animals. They don't like being handled. And I'll tell you what. Okay. There's several things I'd like to show you. So I'm going to come over here because you can't see them. We've got a camera here. Switch to guns. There we go. So, a couple features. Crocodilians have some amazing adaptations. And the first one I want to point out is that you can see these tiny dots on the bottom of their jaw right there. Hundreds and hundreds of dots. Hundreds of dots. These are called dome receptors. And a really good friend of mine named Daphne Soulers discovered these when she was working on a PhD a number of years ago. And what these are, they're pressure receptors. So an alligator can sit on the surface of the water at night in complete darkness. And when something swims by, it makes a series of waves, right? And these waves will come and hit maybe from this angle, and they'll potentiate down this jaw. And there's a series of waves. And so they got 100 eyes on their jaws. They can feel that pressure, sense that pressure change. And what they will do, what they can tell, they can decipher this. Each one of these little dome receptors in the back is conjugated to a nerve bundle which goes straight to the brain. And so basically they can see these things. And they know how far away that animal is, what angle at which it's swimming, how big it is, and how fast it's going, everything, in complete darkness. And she did a really great, you can go to a website and there's a really great little experiment where they have a little 2-foot alligator, they have it in a pen, and it's completely dark. And in this pen, and there's water, and they're filming with an IR, with an infrared camera. And so basically, she'll drip water. And when that drip hits, that alligator, they didn't feed it for it, it turns. It thinks it's something to eat, but it turns. And wherever you drop water, it just lands right on it. She just taped it up, covered up the tape, and then did it. Completely oblivious. Took the tape off, right back. So it's just something simple, but it's a really great adaptation that they can see with their chin almost to some extent. Another adaptation. Let's see. There's several of them. On his back, he's got these osteoderms. Osteoderms are bones that are embedded in skin. And I've got some osteoderm right here. And a lot of people think this is for protection. It certainly is, makes their skin very tough on the back. That's why the back strap right there, the skin is unfit for leather. But what this really is, it is a heat exchanger. Because how do alligators regulate their temperatures? They climb out in the sun. They're ectothermic, they're warm, cold-blooded, and they rely on the heat from the sun. But if an alligator, especially a big one, lies out in the sun, it only heats up the surface. So these little bones that are in their skin have holes in them. And I should have pointed that out over there. Tiny little holes where blood vessels run through. That makes sense because that thing sits in the sun. You've got a whole bank of solar panels that heat up, and the blood courses through there, and then picks up that heat exchanger, and then it goes down into the, where they really need it, where he really needs it, which is in the stomach and the lungs and the kidney, wherever, all the internal organs. So it helps to heat them. They heat them throughout. Let's see, other adaptations. They climb really well. Amazing. Alligators, I didn't really plan on talking about that, but I saw a 3-foot, I have some pens across town where I keep some alligators. It's a hurricane fence. And I saw a 3-foot alligator climb up a 6-foot fence and right over the top. I had I not seen it with my own eyes, I wouldn't believe it. But I saw it. I wish I had film. That would be a great film. What's another adaptation I was going to talk about? They have what is called a nictitating membrane. You can tell how well practiced I am. Look at this. 2 pairs of eyelids. Okay, watch it. The second one is colorless. It's clear, transparent. And this allows, say, an alligator sees a raccoon on the bank, lunges at him and grabs him, chances are that raccoon's not going to go willingly. And it's going to scratch and claw. And what they can do, they can keep their eyes open, but cover it with this pair of goggles and protect their eyes. Because obviously these animals hunt by sight a lot, and they cannot afford to have their eyes compromised. And so that protects the structural integrity of their eyes. In addition, they can swim underwater, and they don't have to worry about, keep their eyes open, see where they're swimming. They don't have to worry about, twigs and leaves and whatever scratching their eyes. So that's pretty good. So that's all. I just wanted to let you meet my friend here. And are there any other questions now, or maybe about crocodilians in general, or? Well, if no, thank you for your time. I appreciate it.